of FDA's three major regulations. Okay, we're going to start off here with slide number 2. And slide number 2, see, these are some of the things that we will cover during this presentation, in addition to any questions that you may have. We define and differentiate between GCP, which stands for Good Clinical Practices, GLP, which stands for Good Laboratory Practices, and GMP, which stands for Good Manufacturing Practice Regulations. We describe how to apply each regulation appropriately. We define the basic founding principles on which each regulation is based, and we discuss the similarities and differences between the three regulations. Slide 3. Good clinical practices, those are the regulations governing human clinical operations. Today, most of the world follows the ICH GCP, the E6 document. That is what FDA recommends that we follow in the United States, and certainly in most of the major regions of the world, this ICH GCP E6 document is part of the law and regulations of the various areas. Good laboratory practice regulations. These are the regulations governing in vitro and animal safety studies. And the good manufacturing practice regulations. Those regulations governing manufacturing, not for non-clinical. Non-clinical being non-human studies, such as animal studies. Clinical, which is human study, and hopefully when you get to commercialize the product for commercial use. Slide 4. GCP, GLP, and GMP are all quality assurance systems. They assure that the operations conducted under each of them, the GCP, the GLP, GMP, are quality operations, and the product or data generated has integrity. Integrity means the product meets its specifications on a reproducible basis, and data generated is verifiable. Slide 5. Let's first talk a little bit about the overview of the drug development process. Today, most companies work in certain pharmacologic areas, for example, cardiovascular, neurological, CNS, and so on. Excuse me. And what the clinical scientists do here is they, first of all, study the diseases in the various areas of the body that they are addressing. And certainly they have databases which they can use, not only the databases on the diseases, not only the information on the product, but they also have some ancillary databases. Remember the Human Genome Project. To find the 20,000 some odd genes in our body, they have that database to work with. And they have numerous other databases which they compile and study the particular diseases in this region of the body. They look at the aspects of the diseases. What does the disease do? How does it enter the body? How does it move through the body, and so on? And then the consequences of the disease. And that is, what does the disease do to the human body? And, of course, there is the major input in which these scientists now are trying to deal with this particular disease or conditions. Of course, I already mentioned number 4, genomics. Proteomics is nothing more than many of these genes produce protein products, such as alpha interferon, for example. Now, what this scientist is going to do is going to take all these databases. He's going to study all these various databases, and then on a computer, model a molecule which can hopefully intercede with this disease process. Now, how can one intercede with the disease process? There's only 4 ways. One, you can develop a molecule that will cure the disease. That very rarely happens. It's possible, certainly is. So number 1 is you can intercede by curing the disease. Number 2, you may intercede by stopping the disease. It will not progress any further. What has been done has been done, but now the disease has been stopped at this point. Number 3 is you can, if you can't cure it or stop it, maybe you can slow it down. So number 3 is slowing down the progression of the disease or condition. And finally, the last one is if you can't do any of the first 3, then maybe you can give the patient some kind of relief, pain relief or whatever. Now, we take this molecule that's been placed on this computer, this single molecule, and we submit it to a process called combinatorial chemistry. What does combinatorial chemistry do? It takes the basic elements of the molecule we just developed on the computer and makes hundreds of versions of it, libraries of it. Now, instead of just looking at one molecule where our success rate would be very low, we are looking at hundreds of different molecules